Hey guys, how are you? Missing all of you. Today our learning objectives are going to be about how to differentiate between complete dominancy and incomplete dominancy, as well as how to make a factorial analysis in the case of incomplete dominancy. To start with, let us differentiate between complete dominancy and incomplete dominancy. Okay, when we studied before in the previous sessions about Mendelian heredity, we noticed that Mendel had studied about the pea plant. Actually, Mendel had studied about 7 traits in the pea plant. Mendel, in his studies, he found that for each trait we have 2 alleles. And these 2 alleles, one of them was dominant, the other was recessive. All his traits, Mendel found this fact. For example, here, for the trait seed shape, he found 2 alleles, round and wrinkled. When he crossed the round with the wrinkled, if you remember in activity 1, he found that all the offsprings in F1, all the hybrids in F1 were round. He concluded that round is dominant or over wrinkled. So what we mean by dominant over wrinkled? Round is dominant over wrinkled, meaning that wrinkled is recessive, it's weak, and round allele is the strong, meaning that when I have the 2 alleles, round and wrinkled, only the round is going to be expressed phenotypically, whereas wrinkled is going to be masked, it's going to be hidden. For all his traits, Mendel found this fact, that for each trait I have 2 alleles, one dominant, the other recessive. And when we came to make factorial analysis for any trait carrying this fact, dominant and recessive alleles, we were symbolizing the dominant with capital letter. For example, the round was symbolized as capital R, whereas the wrinkle, since it's recessive, was symbolized with a small letter with a small w. Also, in all his studying about the 7 traits, we found that Mendel had 3 genotypes, for example, RR, WW, RW. But from these 3 genotypes, we could have only 2 phenotypes, round and wrinkled. Since one of the genotypes, which is RW, which is heterozygous hybrid, it's going to have round phenotype because round is dominant over wrinkled. Keep this fact in your mind because we're going to have some differences when we're going to study about incomplete dominancy. Incomplete dominancy. The scientists who came after Mendel and continued his studies, they found that there exist other patterns of heredity found in humans, in plants, and in animals, where we have some traits that have 2 alleles, but no one of these 2 alleles is dominant over the other, meaning that no one of the 2 alleles is stronger than the other that can mask the presence of the other. In order to understand this fact, let us take an example in plant. The plant that I have chosen for you, we call it or we name it 4 o'clock flower. In this flower, we have for the trait, which is the color of the petals, we have 2 alleles, which are red and white. In nature, we have only these 2 alleles for this trait in this flower. But they noticed that if we're going to cross pure red flower with a pure white flower, The result in F1 is going to be a new third phenotype, which is pink. It's neither red nor white. In the studies of Mendel, in the experiments of Mendel, when he crossed pure red with pure white, for example, it is supposed to have in F1 either 100% red or 100% white. From this result, he could determine the dominant from the recessive. But actually, they found that some traits are following this fact, that when I cross the red with white, a new third phenotype appears, which is pink, for example, in our example about the 4 o'clock flower. They continued the same experimentation as Mendel, meaning they crossed the F1, they let F1 to have self-fertilization, meaning we have a pink crossed with pink. They found upon crossing pink with pink, we have 3 phenotypes in F2, 25% white, 25% red, and 50% pink, meaning I'm going to have 3 phenotypes instead of 2 phenotypes. This is the first difference between complete and incomplete dominancy, which is the presence of 3 phenotypes in F2 instead of 2 phenotypes. Please here you have to pay attention to one fact, that in the case of incomplete dominancy, that we call it intermediary heredity, there is no allele for the pink in this case. Actually, I have only 2 alleles for the trait, which are red and white. But the pink is a mixture, is due to combination between red and white. I'm not going to have pink flower except if I have a cross between red and white. So there is no definite allele for the pink. Rather, I have only 2 alleles for this trait, red and white. Another thing you have to pay attention to is that in the case of incomplete dominancy, the 2 alleles are symbolized by capital letters because no one of them is dominant over the other. For the red, I'm going to symbolize it with capital R. For the white, I'm going to symbolize it with capital W. If you notice that upon crossing red with white, I'm going to have a new phenotype, which is as if I have an intermediate color. I have combination. I have mixture of red and white. Also, we can notice that for the genotypes, I'm going to have 3 genotypes. For example, for the red, I'm going to have RR. For the white, I'm going to have WW. For the pink, I have RW. If you notice, again, I have 3 genotypes. From these 3 genotypes initiated 3 phenotypes, which are red, white, and pink. Also, we have to take care for this fact, which is difference between incomplete dominancy and complete dominancy. Let us explain what we have in this example by factorial analysis in order to understand it more. We're going to apply the same factorial analysis that we applied on the complete dominancy. Here, we're going to start with the first factorial analysis. In the case of Mendel, he had taken, for the parents, he had taken 2 pure lines of colors for the traits. For example, here I'm going to take phenotypes of parent, which are red flower, white flower. So here, the genotypes of the parents are going to be as follows. RR for the red, WW for the white, meaning I started with a pure line as Mendel did. The gametes in this case is the same. I'm going to have 100% of the gametes of the male are going to have R allele, 100% of the female alleles or gametes are going to carry W allele. As a combination between red and white, in the case of Mendel, meaning in the case of complete dominancy, upon crossing the red, pure red with pure white, I have to expect that in F1 I'm going to have either red or white. From here I can determine which allele is dominant, but actually what they have found that upon crossing the red with white, the result was RW 100%, but the phenotypes are going to be 100% pink. Here I can conclude from this, from the presence of a new third phenotype different from both parents. If you notice here, I have pink. And the phenotypes of F1 differ from the phenotypes of both parents, which are red and white. Here I can conclude that this is a case of incomplete dominancy or intermediary heredity. What I mean by intermediate heredity or incomplete dominancy, again, I'm going to explain it here as if I have an intermediate color between red and white, as if I have mixed, combined, or I have blended the two colors, red and white, to give me pink color. So I call it intermediate or intermediate heredity. Then if I'm going to continue as Mendel continued, here when we're going to cross F1 with F1, meaning when I'm going to get in F1 all the flowers having 100% pink flowers, then I'm going now to let them to undergo self-fertilization. Okay, self-fertilization, meaning I'm going to cross the pink with pink. F1 crossed with F1. Remember that all the genotypes of F1 were RW. The gametes of F1 resulting from this cross is going to be as follows. For the male, I'm going to have half of the gametes carrying R allele, half of the gametes of the male carrying W white allele. The same for the female. 50% of the female gametes carrying red allele, 50% of the female gametes carrying white allele W. What would be the result upon crossing these two couples? Table of cross, it's going to explain for me what would be the result. Upon crossing the male gametes with the female gametes, I'm going to have in the offsprings RR 25%, RW 25%, again RW 25%, and finally WW 25%. Here, what would be the genotypes from this table of cross? 25% RR, 50% RW, 25% WW. How I'm going to read these results phenotypically? For the RR, it's supposed to be red, so I'm going to have 25% red. For the WW, supposedly to be white, so I'm going to have 25% of the flowers are going to be white. The difference is here in RW. Here RW is going to show me phenotypically a new phenotype, which is pink, 50% pink. Please focus again that here in incomplete dominancy, from 3 different genotypes, RR, RW, WW, I got 3 phenotypes, red, pink, and white. Whereas in the case of complete dominancy, from the 3 genotypes, we were getting 2 phenotypes. This is a big deal that you have to take it into consideration. You have to keep it in mind. Now, for next time, we're going to explain another pattern of dominancy or heredity, which is co-dominancy, which is somehow like the incomplete dominancy. Okay, till I meet you for next time. I hope that you're going to be fine. Bye.